Hello everyone, good evening and welcome to the week 2 PMRF-NPTEL lecture. I hope I am visible and audible. Someone can, if someone can, yeah. Thank you, Niranjana. So since I think we can wait 5 minutes for everyone else to join. Till then, yeah. We can wait maybe 5 minutes for everyone else to join. Till then, we can go through any doubts you all specifically have about the week 2 lectures in which we discussed genetic engineering specifically, including recombination, restriction enzymes, and PCRs. So, if you have anything particular you want to know about, any doubts that you want to clarify, then we can do that before we start the problem solving, like assignment solving. Okay, so if you have no doubts as such, then we will get right into this week's lecture. So, good evening and welcome everyone to the week 2 PMRF-NPTEL lecture on bioengineering and interface with biology and medicine. PMRF-NPTEL sessions are designed so that a PMRF fellow such as myself can take interactive problem solving sessions with the students who have enrolled for the specific NPTEL course. In our case, it is bioengineering. In this particular session, we will solve the various assignment problems of week 2 bioengineering and try to better understand how to approach the course as well as solve various assignment questions that are given in the course. So let me introduce myself. My name is Anup Chatterjee. I am a PMRF fellow and PhD scholar at the Developmental Neurobiology Lab, Department of Biological Sciences and Bioengineering, Indian Institute of Technology, Kanpur. So in the second week, we have just a second. Okay, what have the picture in this? Excellent. Where was I? So in the week 2, we have gone through genetic engineering basics. Mostly we have gone through what cloning is, how to do the entire cloning procedure as well as restriction enzymes and also the polymerase chain reaction. And we will be solving various problems regarding these only. So if anyone does not have any questions regarding the week's course content, then we'll go right into it. So if anyone wants to ask anything about the week's course content, you can go ahead and ask. Otherwise, we will start with the first question. Okay. So we'll go ahead with the first question today, and we start off right with cloning and restriction digestion. So this is a big question that will have many subparts. So the question is, a PhD student Utkarsh is trying to clone a parasite gene PVX101520 in the PET100 and the PUC19 plasmids by primer-based amplification of the gene followed by restriction digestion and ligation to the respective vectors. Post-cloning, he transformed the recombinant vectors PET100 and PUC19 in BL21 and DH5 alpha respectively, and he does a colony PCR to confirm the successful cloning. Next, he extracts the plasmid from the confirmed colonies and sends it for sequencing. So refer to the image shown below and help Utkarsh to decide which pair of restriction enzymes can be used to clone PVX101520. The options are BTSC1 and DRA1, NDE1 and BAMH1, DRA1 and HIN3, and NDE1 and HIN3. So let us look at the picture that we are talking about. So this is the entire picture. Above we have a linearized PBR3 to I think this the above we have the PUC19 vector with various restriction digestion and other sites present, and below we have the restriction digestion map of PVX101520. And looking at both the pictures, we have to guess which restriction enzyme pair will be best for digesting and cloning this particular gene into the vector. So for a hint so that we can get on with it, uh, the region with a lot of restriction sites present on the plasmid backbone, it is known as the multiple cloning site, and it is there that the cloning will take place. So we will take restriction enzymes that are present here and digest the plasmid with it, and digest the uh, insert that we have with it, and then ligate them both. So we have to choose a restriction enzyme pair that is present from this particular MCS. So just we will have to look through the enzymes that are present here and choose accordingly. So I would like everyone to try and go through the enzymes that are present here and choose the pair that you think is the correct one. Like, you can put your answers in the chat box. I would like you to try and you did get what I was trying to say. You have a restriction enzymes. You have to choose the particular restriction enzyme from the list given here and choose the pair that seems most relevant. Okay, so maybe we'll go through the slides and then we'll try and answer this. So first of the bat, let us know more about what restriction enzyme digestion is. So restriction enzyme digestion takes advantage of naturally occurring enzymes that cleave double-stranded DNA at specific sequences. So the restriction enzymes detect a particular DNA sequence that is known as the recognition sequence, and then cut the dsDNA along at the site known as the restriction site. Those are two different things: recognition sequence and restriction site. So here we have the recognition sequence of the well-known restriction enzyme EcoR1, which is GAATTC, and the restriction site lies inside it right after the first G's. One other thing that we have to like look at here is that restriction enzymes always detect palindromic sequence of palindromic DNA sequences, which means that the restriction site for EcoR1 is GAATTC, and in the other strand we have GAATTC but from the reverse side. This is known as a palindromic sequence. So restriction enzymes recognize these type of palindromic sequences. So there are hundreds of different restriction enzymes which allow scientists to target a variety of recognition sites. Restriction enzymes that are used mostly in lab are type 2 restriction enzymes that recognize palindromic DNA sequences and cleave the dsDNA, double-stranded DNA, at the recognition and restriction site. So for type 2 restriction enzymes, The recognition site is the restriction site. So we have very well controlled DNA restriction enzyme which can cut DNA at the exact site that we need. And that is why type 2 restriction enzymes are most used in labs. So restriction enzymes can produce sticky ends or blunt ends. What I mean from that is that what I mean by sticky ends is that here when you see the cleavage by EcoR1, there are overhangs of single-stranded DNA. In the upper strand we have AATTC and the lower strand we have TTAA. We have an overhang. So these overhangs, when you place these two sequences close together, the A will form a hydrogen bond with T, this A will form with this T, and these two with these two T's. And thus, there is a hydrogen bonding interaction that brings the double-stranded DNA close together again, which helps in ligation. So sticky ends are compatible sticky ends. So sticky ends that can form complete base pairing with each other are known as compatible sticky ends. And these help in ligation and can be easily ligated. But if we have a enzyme which cuts blunt end, like from the middle of the sequence, so we have no overlap. So we have one the cut right here, bit after the GAA and before the TT. So a cut right here will produce no overhangs, 3 nucleotides here and 3 nucleotides here. And these sites are very difficult to ligate, but are compatible with all other blunt end restricted sites. Because as you can see, there is no question of base pair. So blunt ended ligation is difficult, but all blunt ended ligations are compatible with each other. So this was about restriction enzyme digestion. So does anyone want to ask anything about this? You have gone through restriction enzymes before, right? Anyone? Hello, am I audible? Yeah, okay. So no questions on restriction enzyme digestion as of yet. Yes, no. Anything will suffice. So I will assume that it's all fine. So we move forward with cloning with restriction enzyme digestion. Yeah, we have a question. Arushi asks, what is the difference between compatible and blunt sticky ends? Okay, so maybe I have to explain this again. Let me try. We have, let me draw and try how. We have a white bit. Okay, so take yours. My new screen is visible, right? Yeah. So what I mean by compatible is, say you have a DSDN strand right here, and what a You can either digest it with a restriction enzyme which produces sticky ends. So say the example we had GAATTC, and here we have the same thing but opposite order GAATTC. So this is my upper strand. Enzyme like EcoR1 will cut right here and produce something like this. So this is known as a, so we have a 5 prime here, 3 prime here, and here we have a overhang. So this is known as the overhang, and this is what produces the sticky ends. So this is a sticky ended layer or digestion. So these are the sticky ends. Now, what I mean by a blunt ended digestion is we have something like this. Same thing, and the digestion occurs right down the middle of it. So when the digestion occurs right down the middle of it, the two strands that we have are like this, with every one of the bases already base paired and no overhangs present. This is known as a blunt ended digestion. Now the difference is when I want to get a ligation done between two fragments that have been digested with equal. So instead of this fragment right here, if I had another fragment with the exact same digestion, then I will still have this overhang present. And any other sequence that has been digested with eco-R1, when I put together with this 3' end, will base pair like the original DNA will, like the T's with the T's, the A's with the A's, and then it will form a nicked structure such as this. So in which everything has been base paired, but there in this end we still have a phosphate, or rather a OH. So we have a nick present still here, which can then be ligated. So what this does is this helps in the ligation process by bringing the two DNA strands close together. But in case of blunt cloning, there is no scope of hydrogen bond because everything is already hydrogen bond. So what the ligase can do is that in case both of these come together, it will just form a bond between these two. And thus, if we have two sequences that have been like it restricted digested by equar1, then they are compatible. That is, they will form hydrogen bonds between them like this that we have seen here. But instead, for blunt-ended cloning, there is no question of compatibility because there is no hydrogen bonding that is going to happen. So we will just have the ligase bringing together two separate pieces of dsDNA and ligating them. And as you can see right here, since there is no hydrogen bonding, compatibility is not an issue. So if I, instead of GGGCCC, we had something else like AATTAA instead of this fragment, this can also go and ligate to this without any issue of compatibility. So this is what I mean by compatible ends. Is that okay? Yeah, so I since we are here, I will want to go and try something, like add something. So say you have a say you have a dsDNA which looks something like this, which is known as an insert, and you want to place it in another vector. So another vector that looks like this. So this is a ds vector, so we have two strands, and here you have restriction sites for two different restriction enzymes such as ecoR1 and HIN3. Say what you will do is that you will digest your insert with ecoR1 and HIN3 as well. What this gives you is two separate DNA segments which look something like this. So this end is ecoR1 digested, this end is HIN3 digested. Now what will happen is that when you digest the vector as well, the vector will also have two sides. One is here which is ecoR1 compatible and one here which is HIN3 compatible. And when you place both of them together, the entire thing can form a covalently closed structure such as this, where this is the vector backbone, which will then go ahead and base pair with our insert. And now ligating is very easy. Just the ligase will form the bonds right here, and we will have a circular plasmid with our insert inside it. So bonding will be done, and we have our insert inside the vector backbone. And this only happened because our when we digested, we used two enzymes which were compatible with the enzyme that a vector was digested with. This entire thing is known as restriction cloning. Is that okay? Did we get all that? Yes, no, you can reply back and tell me that you did not get anything or you understood. I will discuss this in the later slide as well. Is this okay? Hello. Yes, thank you. So, okay. So let us look at. So the PPT is visible now, right? The PPT is visible. Okay. So let us get along. So that was the restriction enzyme digestion, and now we will look into restriction enzyme digestion, cloning with restriction enzyme digestion. This is something that just I just draw to you. And what we have is that we have a particular insert, which is our gene of interest. And what we do is that we digest it with two separate enzymes, which in this case is ecoR1 and not one, which will give us a insert fragment, which looks something like this. Next, we'll look at our vector where we want to put this insert into, and we will also restrict it using our same enzyme combination. So we have the ecoR1 and not one ends present here, and we will ligate both of them. So this will go inside here, and ecoR1 ends will form base pairing with ecoR1 end, and not one will form base pairing with not one. And when we ligate it, we will have a complete circular DNA which is ligated. This is cloning with restriction enzyme digestion. So what more we can do? Say in case you have, so as you know, just as we just discussed, restriction enzymes have a specific DNA sequence that they recognize. What if the DNA sequences that your vector has is not present on your insert? So say you had your eqr1 and not one sites present on your vector, but your insert did not have those sites. So how will we ligate both of them? What we do is we amplify our particular insert using primers and PCR, and our PCR primers have these particular sites present in them. So they will have the eqr1 and not one sites present in them. And since the primer is external DNA, this just adds some bases to the ends of our insert, which doesn't hamper the insert as such, but just adds the restriction sites on both ends. And then we can restriction digest and then like it to form the vector present inside the insert. So this was cloning with restriction digestion. And here we are back to our question where we have to choose a pair of enzymes which will digest our vector backbone and help us ligate the the insert of our choice into the vector backbone. So can we now choose the restriction enzymes present here and match them to the options directly? So does anyone want to try and do this? Like just choose an option. Out of A, B, C, D, we have four options which are pairs of restriction enzymes. Okay, so if we look at the pair of restriction enzymes given here and compare it to the multiple cloning site present in the question, we will see only two enzymes match the given MCS, which is the ND1 enzyme and the HIM3 enzyme. So the correct option is number 4. So is that okay with everyone? Okay, so we can go forward to the next question. The next question is, Utkarsh again was given a vector of overall 20 kbs to perform a restriction enzyme digestion. After single and double digestion of two enzymes A and B, he obtained the following gel image, which is like something like this. So first he digested with restriction enzyme A and he loaded here and he saw two bands. With restriction enzyme B and he saw one band. But when he digested with A and B, he saw four bands. So now the question is, given the following information, ascertain what information can be obtained about the restriction sites of A and B in the vector. The options are A has 3 sites and B has 2 sites on the vector. A has 2 sites, B has 0 sites. A has 3 sites and B has 1 site. And A has 4 sites and B has 1 site. So this is a single correct type of answer. You can just go ahead and type it in, whichever you think is the correct answer. Arushi says it is number C. A has 3 sites and B has 1 site. Okay, anyone else? Okay, so let us try and solve this. I think I will have to draw this one again. Yeah, the screen is visible again. So what happens with this type of question is that you first have to understand that how many fragments can be created with the restriction enzymes acting on a circular vector. So in case the vector is circular, let's say it looks like a circle. So we have something like this, and if there is one restriction site of A present here, then we will have one double-stranded linear DNA. That is, this will mean one band on the gel. If say we have two sites, this will mean two bands. This is because this will form one band from here to here, and this will form the other band from here to here. This is how you form two bands. Similarly, if we have one more, then we will have three bands. So if this time it will be from here. So this means in case of a circular vector, the number of restriction sites equal to number of bands. Unless two bands are of the same size. Say if this band, say band A and band B was of the same size, then on the gel we will only observe one band. So we will only observe one band because both of the DNA sizes are the same and they will overlap. So unless they are of the same size, we will see two bands. So if they are of different sizes, we will see different bands B1 and B2. But the basic remains the same, and the number of restriction sites in a circular DNA, the number of bands. So depending on that, in the given question, you can see that A. So the PPT, is the PPT still visible? Is the PPT still visible? I don't think so. I think. Hello. Yes. So we have well, picture. So in the given question, we have A that forms two bands and B that forms one band. So from our discussion, it is obvious that A will then have two restriction sites on the DNA or the vector, and B will have one site. And this, this is what happens. And the wait a minute, someone has to. So A forms two bands seemingly on the gel, and B forms one band. From only this information, it looks as if has two sites on the DNA and B has one. But when we digest both A and B, we see we have four bands. And we see that the thick band A had, the thick band that A had here at around 6 kb, that also gets resolved. So this band goes down and we have two thinner bands in its place. And B only forms again one band. So all of this information together tells us that A might have had three restriction sites and two of the bands overlapped and form a thicker band, whereas B only had one site. And when both an A and B are used to restriction digest our vector, we have four bands in total because this particular band is disrupted. So the correct answer is A has three sites and B has one site in the vector backbone. Is that okay? Probably not. Is that okay with everyone? Did everyone understand why we say ki A had three bands and B had one? Okay, Arushi says yes. So Sharan also says yes. Okay. So we'll move forward with the next question, which is a continuation of this restriction digestion problem. And this is a bit difficult. So I would ask everyone to use your pen and paper to solve this one. This is one of the basic questions that comes from this particular ATM. So PUC1 vector is digested with EcoR1, which resulted in a band of 26 kb. Band H1 digestion resulted in four bands of 13, 6, 4, and 3 kb. When both the enzymes were used, it resulted in four bands of 7, 6, 4, and 3 kb. Develop a plasmid map and show the restriction sites. So we go back to the drawing board and draw the entire scenario. Not essentially draw, but write it down. We can solve it by writing it down. So we have the PUC1 vector, and it was digested using three enzymes. First was EcoR1, and it formed a single 26 kb band. Next we had BAMH, which formed four bands: 13, 6, 4, and 3. And finally we had EcoR1 plus BAMH1, which again formed four bands only: 7, 6, 4, and 3. And now the question is if we can develop a plasmid map. So plasmid map means that just to draw circular plasmid, circularish. So this is my PUC1 vector, and you mark your restriction sites on the vector. So let us go ahead and try this. Does anyone want to try and tell me how exactly you can do this? You can unmute yourself and speak to the mic and tell me how exactly we can go ahead with solving this problem. Hello. Okay, so we'll go ahead and try solving this problem together. But you see, these sessions are meant to be interactive in nature. So it is way easier if everyone tries and interacts and tries to we solve this together. Because otherwise, this will only be a recorded video that you can watch on YouTube. No reason of joining the session itself. So first off, we do the easier work, which is this we can solve in two ways. One is the sequential method, where you first take your vector and first put one, like one by one, you try and put both in the restriction site. So I will put our eco R1 site right here. So eco R1 site, there is one eco R1 site because there is only one band, as we just discussed. Circular vector, one restriction site, and one band. So eco R1 only cuts once the entire vector. So we have something that looks like this. Next we have BAM H1. So BAM H1 can be anywhere, and since there are four restriction bands, there will also be four restriction sites. So in case of BAM H1, we can again draw four different restriction sites. One can be here, one can be here, one can be here, and one can be here. So these are the BAM H1 sites. B site, B site, B site. This entire thing is huge, and this will be a 13 kb band. And then progressively we have smaller bands. So we have this as the 6 kb, this as our 4 kb, and this as our 3 kb bands, which solves the BAM H1 problem. Now we have eco R1 that can be present anywhere, but the given information tells us that when both eco R1 and BAM H1 were used, we still have four bands present. That is 7, 6, 4, and 3. So what this tells us is, referring back to our last problem, that what happens may be that there are actually 5 bands, because obviously eco R1 will cut at a separate site from BAM H1 and form another band. So we should have had 5 different bands, but we have only 4. So how exactly to tackle this? We can assume that the 13 kb fragment that we had after eco R1 digestion, it formed a 7 kb fragment and a 6 kb fragment. And when we run it on a gel, the 6 kb fragments from both the digestions overlap. And thus we only see 4 bands, but actually there are 5. 4 from the BAM H1 digestion and 1 extra from the eco R1 digestion. But since the weights overlap, we only have 4 bands. So equipped with this information, we can safely say that our eco R1 site is in middle of the 13 kb fragment, which cuts the 13 kb fragment to a 7 kb and a 6 kb fragment. Thus our entire restriction map should look something like this. BAM H1, which will have 4 sites, and eco R1, which will have 1 site present inside the 13 kb fragment of BAM H1. So that is what we have here. That is what we have here, that eco R1 site is present in the 13 kb fragment between two BAM H1 sites. So this is our 7 and this is our 6 that we just drew. So this is what the plasmid map will look like. Is that okay with everyone? So we have a, yeah, Arushi says it's okay. Anyone else? Does anyone have any question regarding this particular one? So BAM H1 we have a 3, 4 and 6 fragment. We should have a 13, but eco R1 comes in and cleaves it into 2 into a 7 and 6. Two sixes mixed together to form a single band, and thus we only see 4 bands as given in the question. So this was the problem on restriction digestion. Now we move on a bit and change our pace and go to a question about PCR. So Utkarsh performed PCR and has kept his samples with master mix properly set up in a thermocycler instrument. He had to set up the process himself owing to no available preset protocols. And here is what he set up. He set up 5 steps. The first step being heating the PCR reaction at 94 degrees Celsius for 3 minutes, followed by a 94 degrees Celsius for 30 seconds, followed by a 72 degrees Celsius heating at 30 seconds, and the entire program will go back to the second step. That is, it will cycle between 72 and 94 30 times, followed by 72 degrees Celsius for 10 minutes, and then holding the entire solution at 4 degrees for 60 minutes. After running the agarose gel, he finds that there was no amplification. Which of the following could be reasons for this problem? The options are: everything is fine and products will be obtained as expected; denaturation of the parental DNA did not take place; extension step did not take place; and annealing of the primers with the template did not take place. So these are the 4 options that we have. So you can go ahead and put whatever you think is the correct one in the chat box. Why do you think that there was no amplification found? Sharan and Arushit both say that it is D, annealing of the primers to the template did not take place. Okay, so let us look into what exactly PCR or polymerase chain reaction is before going forward. So polymerase chain reaction is a revolutionary method developed by Carey Mullis in 1980s and is essentially in vitro DNA replication. PCR is based on the using the ability of DNA polymerase, which is specifically the Taq polymerase, isolated from Thermus aquaticus, that can withstand high temperatures, to synthesize a new strand of DNA complementary to the offered template strand. Because the DNA polymerase can add nucleotide only to a 5' pre-existing 3' OH, it needs a primer to start amplification, and by the end of PCR we will have billions of copies of the given template that we started with. So essentially what happens is that there are three separate steps to the polymerase chain reaction. The first step is denaturation, in which the double-stranded DNA under heating condition denatures or dissociates into two single strands, which looks something like this. This is followed by primer annealing, in which our given specific primers, which is the forward primer and a reverse primer, attach to the template strand, one to the upper strand, one to the lower strand. And what this does is, this will start the amplification in the primer extension step, which is at 72 degrees Celsius, which is the temperature at which Taq polymerase works. So this is the optimal temperature for the enzyme, and it starts adding nucleotides towards the 5' end, and we have amplification, as we can see right here. So after one cycle, we will have from one strand of template, 2 strands of templates, and will look something like this. And this is then continued or repeated 25 to 35 times to form millions of copies or billions of copies of DNA. Since this whole process is exponential, by 35 times we will have 2 to the power 35 number of DNA copies, which is a lot. So essentially we are amplifying our prior given template using the entire process. So the required steps for the PCR reaction include a DNA template like this, a DNA polymerase, which is the Taq polymerase that we mostly use, specific DNA primers, which will amplify the DNA region of our choice, which will lie between the two primers right here, DNTPs or deoxy NTPs, deoxynucleotides, which will actually be added here by the DNA polymerase, and a thermal cycler, which is the device that is used to maintain this varied temperatures 96, 55, and 72. And the steps of PCR are denaturation at 96. The high temperature is required in order to melt the DNA into two single strands. Primer annealing at 55, which is a very low temperature, close to the melting point of the DNA so that re-annealing or zipping back of DNA can happen and primers can go and form the hydrogen bond. So here you are breaking the hydrogen bonds between the two big strands, and at this temperature you are allowing the smaller primers to go and bind and form the hydrogen bonds again. And then at extension step, the DNA polymerase functions and adds the nucleotides, and we have extension of the template to form something like this. So this is entirely the polymerase chain reaction. So does anyone want to add anything to this? Any questions regarding PCR? Oh, so let me ask instead, or ask a question to you instead. Why do you think that a normal DNA polymerase, which we have, cannot be used in the PCR? We need the TAC polymerase, which only acts at such a high temperature. Why do you think that is the case? Can anyone tell me that? You can write your guesses into the chat box also. Like you can just go ahead and guess what could be the reason why we need a TAC polymerase or the temperature, like polymers that can survive at that high temperature. Yeah, so Sharan says because high temperature is attained and normal polymerase cannot survive. And Arushi says DNA polymerase is unstable and gets degraded on heating. That is correct. That is absolutely fine. That normal DNA polymerase will get denatured and cannot like non-functional at that high temperature. But this begs the question that why is the high temperature required? So why can't this be a 96 degrees Celsius denaturation step, 55 degrees Celsius annealing step, and a 37 degrees Celsius extension step, which can be done with any other polymerase that we have? Why is a high temperature of extension required? Like when you take a high temperature of extension, you need a temperature independent polymerase such as stack poly. But if you do not, like, why is the temperature necessary in the first place? Can we have this at 37 degrees Celsius or room temperature and like use normal DNA polymerases? What do you think will be the, like, why don't we have the primerase extension at 37? The extension step needs this higher temperature at 72 degrees Celsius. No one has any guesses. Like you can put in your guess, like whatever you think can be the reason as well. Okay, so primary extension occurs at 72 for the very logical reason that primer annealing or the melting point of DNA, double-stranded DNA, is around 55 degrees Celsius. If we go way lower than that, like around 37 degrees Celsius, the entire strand will be closed up again. So it will again form base pairing and will form double-stranded DNA instead of single-stranded DNA like here. And double-stranded DNA, like our DNA polymerase, will not act on double-stranded DNA. So essentially, after primer anneals, if you lower the temperature, the entire middle portion and the side portions of the double-stranded DNA will also base pair, and this will form a closed structure, and DNA polymerase cannot go in and start extending the entire template. So we will only have these two copies which are awkwardly double-stranded in our last mix. So no extension will occur. So this is why the primer extension should occur at a high temperature such as 72 degrees Celsius, where no annealing of the primary strands can take place. So the big strand that we have, the upper strand and the lower strand, do not base pair back. That is why a high temperature such as the say, like the one that is given is required. Is that okay? Right? Yes, no, maybe. Okay. Can I explain it once? Next. Okay. So what happens in all these steps? So in the first step, that is denaturation, we have two single-stranded DNA strands. In the next step, annealing, the primers goes and binds on the single-stranded DNA strands. Now, this single-stranded DNA is required for the replication to take place. So our polymerase can only bind to the single-stranded DNA on one end and the primer to the other, and then start the template new strand synthesis. If in And that is why we need a high temperature so that these remain as single strands. If we lower the temperature from 55 to 37, what will happen is that re-annealing of the two strands will take, so they will go back to this original state, and in this original state the DNA polymerase cannot access the DNA information, like it needs to bind to the single stranded DNA. It cannot bind to the double stranded DNA, and once it goes back to the original stage at the lower temperature, amplification will not occur, and that is why we need a high extension temperature so that it remains in single stranded states. Is that okay? So am I audible now? Hello, you can hear me, right? Okay. So that was that. So that was PCR. And we can go back to our question. So what Utkarsh did wrong here is like you can see it once we know the steps of PCR amplification. So first we have the 94 degrees Celsius for 30 seconds. This is the denaturation step. And then we have 72 degrees Celsius for 30 minutes, which is the extension step. There is no annealing step present inside, which should be around 55 degrees Celsius. So no chance for the primers to go and anneal and then start the DNA synthesis. And that is why there is no amplification. So the correct answer is annealing of the primers with template did not take place, and that is why there was no amplification found. So our correct answer is D, as was given in the chat. So anyone wants to ask anything regarding this? Okay. So if not, then we can go forward. So refer to the hypothetical sequence of the PVX101520 given below and help Utkarsh to design a pair of forward and reverse primers. Choose the correct pair of primers. So we are asked to design the primers that we were talking about just a bit before. And primers are obviously complementary short strands of DNA. And we are asked to design the primers for the two strands given here. So the reverse primer will go and bind here, and our forward primer will go and bind here. And we are given options for the primers. 1, 2, 3, 4, and we have just to choose what the primers are. So you can go ahead and like try and solve this and tell me what the primer sequences should be. Just they will be just complementary to the original sequences. So like if you just go ATGC and go back, your back calculate, you will find the template sequence, the primer sequences. So you can put that into the chat, whichever you think is the correct answer. Okay, so let us try and solve this instead. So what exactly are primers? Primers are short single-stranded DNA fragments which are used in several cell laboratory techniques such as PCR. In PCR, a pair of primers hybridize with the sample DNA, which defines the region to be amplified and results in a copy or millions of copies to be formed, just like we saw in the previous question. Primers are necessary for DNA strand polymerases to synthesize a new strand, and primers are essential for DNA polymerases because DNA polymerization has to have a starting primer. Without a primer, DNA polymerization cannot start. In vivo, an RNA primer is synthesized by the primer's enzyme during DNA replication. So, in normal conditions inside the cell, the primer is generated by the enzyme primase, which begins the DNA replication process. But in laboratory condition, we do not use a enzyme such as primase. We instead make our own DNA primers, which we can synthesize. So say you have a template DNA with something looks something like this, and you have your region to be copied. What you do is you like specific to your region that you want to amplify, you will form a primer corresponding to this. So you take a primer and that will bind here. So from the TGAGT sequence, you take the complementary one. So you take a ACTA sequence as your reverse primer, and a forward primer will be the complementary sequence TATCAG, which will bind here. And then you let the PCR run, and it will amplify your particular DNA. So this is how the first primer will look like, which will be exactly identical to the opposite strand, and this primer will be exactly opposite to the identical strand. And this will help you with the PCR process. So given the particular information, what we will have to do is just find the complementary to this, so which is the lower strand itself. So the reverse primer sequence will be TAGAT, TAGAT, and the forward primer sequence will be AGGTCAT, and it is AGGTCAT, which is the second option, which is the correct one. One thing you have to keep in mind is that ordinarily, like the DNA sequences are traced from 5' to 3' direction. So reverse primer, even though from when reading left to right, it should be GCGATA, the sequence should be. But this is when you are reading it from the 3' to the 5' direction. You will have to write it from the 5' to the 3' direction, that it will start from this end. This is a common mistake that is often done. So do not fall into that trap, and thus the correct answer is the second one. Is that okay? Hello? Okay, so we move on to the next question. So we come back to Utkarsh, and Utkarsh ran a agronomist gel. which is used in the DNA electrophoresis method, which is used in the DNA. So Utkarsh ran an agarose gel. Hello, Divan, do you have anything to speak? Okay, okay, so problem. Huh? So we go back. So Utkarsh ran an agarose gel to check the quality of the extracted plasmid, but found on the gel run blotches such as this. So what can be a possible issue with the entire problem? So electrophoresis is the technique which is used to detect the size of the DNA. So you load your particular DNA sample in these lanes or wells and DNA and attach a negative electrode here and a positive electrode here, and the DNA will run from a negative to positive end. But you should see sharp bands in your DNA gel. But what Utkarsh is looking at is blotches such as this. So what can be a problem which actually gives rise to this? So the options are ETBR is not well mixed, gel was run from the positive electrode to the negative electrode, gel was run from the negative electrode to the positive electrode, and wells are not properly made. What do you think is the problem that is occurring here? So you can go ahead and type in the chat box which you think is the correct answer. Okay, so we'll try and solve this. So this is the gel electrophoresis method. What we first do is that we form the agarose gel, in which we first use something known as a cast, and we pour our agarose solution into it, which is heated at a high boiling temperature to mix the agarose, and when it cools down it will form a gel like structure, and we will then have a solidified agarose gel in which we form the wells using a ladder such as this, and which will form essentially holes or square wells present on one end of the gel, and then we load our DNA solution into it, and after running using an electrophoresis apparatus, the negative end towards the well side or positive end towards the other side, we will see the DNA migrates, and after some time we will see sharp bands such as this. So this is how gel electrophoresis works. It separates DNA based on their weight. And the in case so you will problem is that you will not be able to see DNA with your naked eye. So in order to look at DNA, we use a fluorescent compound known as ETBR, and it is put into the agarose gel or stained. The agarose gel is stained with ETBR after the gel run, and what it does is it binds to the DNA and then fluoresces orangish or pinkish under orangish under uh, orangish or pinkish under the UV rays, and we will see a image such as this. But blotches which look like these occur especially when ETBR is not well mixed to the DNA, and we will have not like our bands will not be sharp at all, and we will see smudges throughout the entire gel. So this was with Utkarsh's problem here. Next is Utkarsh went ahead and reran the gel, and after running the gel, he found that the gel looks something like this. He found a uniform layer of DNA. So this everything that is pink marked is DNA. So he formed a uniform layer of DNA. So again he has this problem with his gel, and he asking what can be the problem. So the options that are given are ETBR is not well mixed, gel run from positive electrode to the negative electrode was run from the positive electrode to the negative electrode, gel was run from the negative electrode to the positive electrode, that is opposite to the correct one, and wells are not properly made. Which do you think is the problem this time around? You can post your answer in the chat box. What do you think is the like what is wrong with this particular gel? Sharon says it is D. Wells are not properly made. So you are correct, Sharon. The option is D. The wells are not properly made. This happens when the boundary between the wells are breaks. So what might have happened is when Utkarsh went ahead to load the gel, the wells were not properly formed and they were essentially connected with each other. And so the DNA mixed from 1, 2, 3, and 4 all mixed together, and instead of having distinct gel DNA runs in each of the lanes, we had one uniform gel run throughout, and we had one uniform DNA front, which looks something like this. You are correct. So we move on to the next question. What are the major steps for gene cloning via vectors into bacterial cells? So we learned about cloning, about the restriction digestion part of the cloning before, and now the question is, what are the major steps for gene cloning via vectors into bacterial cells? The options are competent cells preparation, followed by providing the insert, providing the vector, and provide restriction and ligation enzymes. The next option is restriction enzyme digestion of vector, transferring it to the competent cell, restriction enzyme or digestion of the gene separately, and transferring it to the competent cell next. So first to digest the vector, then transfer it to the competent cell, followed by digestion of the insert, and followed by transferring it to the competent cell. Third option is restriction enzyme digestion of the vector, followed by restriction enzyme digestion of the insert, ligating the insert and the vector, and transformation of the recombined vector to the competent cells. And finally, we have restriction enzyme digestion of the vectors, transfer it to the competent cells, restriction enzyme digestion of the insert separately, and transfer it to a different competent cells and fuse the two cells using ligase enzyme. Okay, so the only one of the four options are correct. You can try and answer this in the comments, like put the answer into it. Okay, so let us try and understand what exactly gene cloning is. Gene cloning is the method in which you try to make multiple copies of your gene by various. So the normal way what we do is we take our gene, put it into a vector backbone, and put the entire recombinant DNA molecule into a cell and allow the cell to proliferate. Since the cell carries the plasmid and your gene of interest and it proliferates in large numbers, will have at the end of the day large amount of your gene. So your gene is cloned. How is it done? First, you do what you do is you take your plasmid and you isolate your plasmid, and as we had just learned, you digest the plasmid with a restriction enzyme. You take your particular gene of interest and also digest it with the same compatible enzyme, and then combine the targeted fragment and the plasmid DNA. So exactly like we just saw in one of the previous questions. After combining, you will have sticky and compatible sticky ends base pairing together, and then you ligate add DNA ligase and ligate the recombinant DNA plasmid. So you will have a circular plasmid having your gene of interest. Next, what you do is you put the plasmid back into the bacterium by transformation. And what transformation is essentially putting your plasmid of interest into your bacterial cell. You put your recombinant DNA plasmid into your bacterial cell, forming a recombinant bacterium. And since bacteria proliferate at a huge rate, you allow it to grow. And after bacteria reproduce, you have multiple clones of cells which will contain multiple clones of your genes. So this is how gene cloning works. Any questions from this? I guess not. So we can go ahead and try and answer this question. So it is number C, restriction enzyme digestion of the vector, followed by restriction enzyme digestion of the insert, ligation of the two, and then transformation into competent cells. So restriction enzyme digestion of the vector, restriction enzyme digestion of the gene, ligating both of them, and then transformation into a vector. That is the answer. So we move forward to the next question. Heat shock method of transformation is done to transform the recombinant vector in the host system. What is the type of competent cells required for the heat shock method? So we just talked about transformation. So transformation has one or two methods that can be used. Mostly used are heat shock calcium chloride method and the electroporation method. Here we are being asked for the heat shock method of transformation. What kind of competent cells do we need? The options are these are 10% glycerol competent cells, calcium chloride competent cells, naturally competent cells trained for the used vector, and competent cells are not required in the transformation. So only one of the options are correct. I would like everyone to try and put your what you think is the correct option in the chat. So only one of the options is correct. Okay, so let us try and understand what competent cells exactly mean. So, non-competent E. coli cells are E. coli that have not been made competent. And what that means is that they will not take up plasmid DNAs that are introduced into the environment. So, any DNA molecule that is going around the cell, it is not viable for the cell to take it up. So, it will not. So, any plasmid that is going around or introduced in the vicinity of the E. coli will not even care about it and will not take up the given plasmid. But on the other hand, competent E. coli cells are made competent either using the calcium chloride heat shock method or the electroporation method. And what it does is that it makes the cell viable for taking up the plasmid. Essentially, it makes the E. coli cell membrane permeable, making various pores via which the plasmid can enter. So this is what we mean when we say that a cell is a competent cell, that it is able to take up any plasmid or other DNA that is injected or present in its vicinity. So competent E. coli cells are majorly used in gene cloning experiments because these are the best cells for taking up the plasmids that we want to introduce into them. So how exactly are competent cells made? So we will be discussing the calcium chloride treatment method. So ordinarily, we know that DNA is negatively charged because DNA has a phosphate backbone which gives it an overall negative charge. Whereas our cell membrane, the plasma membrane, is also negatively charged because it has various phospholipids. So obviously the negative charge of the DNA and the negative charge of the plasma membrane try and push each other away, and there is no possibility of the plasma even getting close to the cell. So how is this solved? What we use is the calcium chloride method. We treat the cells and everything around it with calcium chloride. Calcium chloride obviously has calcium in it, the Ca2 plus ion, and Ca2 plus ion goes and neutralizes the negative charge present all over the membrane as well as the DNA. What both of these do is that calcium from the CaCl2 interact with the negative charges present here, which leads to an electrostatically neutral environment. So the entire environment gets neutralized and our cells can easily take up the plasmid because there is no electrostatic repulsion between the plasmid and the membrane anymore. So the plasmid will go ahead and get right through the plasma membrane. This is what we mean by competent cells. So the heat shock method of transformation is done to transform the recombinant vector in the host system. What is the type of competent cells required for the heat shock method? The answer is calcium chloride competent cells. So that is that one. So we move on to the next question, which is which of the following methods explains the correct principle of heat shock method of transformation? So we just talked about the heat shock method. Where does the heat shock come in? What exactly do I mean when I talk about the heat shock method? And what exactly does it do? So the options are it halts the reactions inside the cells to let transformation happen without any hindrance, but to lyse the cell's protective layer, cell membrane or wall, to loosen the protective layers and let foreign objects go inside the cell, and to digest the cell's protective layers and let the internal components come. So only one of the following options are correct. You can go ahead and try putting the correct answer into the comments, which you think is the correct one. So what does the heat shock do? It halts the reaction inside the cells and let transformation happen without hindrance. It lyses the cell's protective layer, which can be the cell membrane or cell wall. It loosens the protective layer and lets foreign objects go inside the cell. It digests the cell's protective layers and let internal components come out. Which do you think is the correct option? Okay, so let us try and answer this. What is heat shock? So calcium chloride competent cells are also known as chemically competent cells. So as we only just saw, initially DNA and plasmid and membranes are repulsive due to presence of negative charges. So the plasmid and the DNA membrane wants to go far away from each other. But once calcium chloride treatment is done, calcium chloride coats the negative charges and neutralizes it. So this causes the plasma to come close to the cell, but still the plasma membrane does not allow the DNA to go in. So what we do is that we give it a heat shock. That is, we allow it like it is ordinarily at lower temperature. We suddenly increase the temperature, and this sudden increase of temperature, what it does, it forms pores on the membrane. So essentially, holes present in the membrane that through which the DNA now can go right into the bacterial cell. So holes are formed by the heat shock, and it goes right in. Essentially, the heat shock step lyses or produces pores or holes throughout the protective layers of the cell membrane or the cell wall, which allows the plasmid DNAs to go inside. So the correct option is, which of the following methods explain the correct principle of heat shock method of transformation? The options are, and the correct option is to lyse the cell protective layer, cell membrane or cell wall. Okay, so we go on to the next question, which is a vector was taken as the cloning vector for inserting a gene of interest, GOI, having a streptomycin resistant gene of its own. The HIN3 cut site is present in the penicillin resistant gene present in the vector, and no other antibiotic resistant genes are present in the vector. HIN3 restriction digestion was performed to insert the gene of interest into the vector. The resultant vectors, after complete transformation in the competent cells, devoid of any antibiotic resistant genes of its own, our desired recombinant cells were obtained. Which of the following is the desired transform? So, so what happened is that, uh, as we just saw, that the vector is ordinarily cut with a restriction enzyme, and our insert is also cut with a restriction enzyme. What here, what the case is that when we are going to cut the vector, there is only one antibiotic resistant gene present on the vector, which is the penicillin gene. But uh, during the cutting of the vector using the HIN3 enzyme, the penicillin gene itself gets disrupted, and using that digestion, the gene of interest is cloned into the vector. But on the another hand, the gene of interest itself has a streptomycin resistant gene. So now the question occurs is that if we plate these transformed cells, which of the following will be the desired transform? The options are, the desired transformant will survive both in penicillin and streptomycin, which are two different antibiotics. It will survive in penicillin and not in streptomycin. It will not survive in penicillin nor in streptomycin. And it will not survive in penicillin but survive in streptomycin. So you can go ahead and put what you think is the correct option in the chat box or comments. Okay, so we'll go through bacterial selection using antibiotic resistance. So what is bacterial selection? What happens is that plasmid vectors carry specific antibiotic resistant gene. So say you have transformed your particular bacteria that you had with the plasmid. But how will you know which bacteria colony has taken up the plasmid? What you do is called selection. Our bacterial plasmid that you wanted to transform into the cells has a antibiotic resistance gene. Now, these genes allow the researchers to detect whether the bacterial cell took up the plasmid with the gene of interest or not by plating the bacteria on the media containing that specific antibiotic. So say your particular plasmid had a penicillin resistant gene. So if the bacteria has taken up the plasmid, only then the bacteria will be. Hello. So I'm probably audible and visible now. I lost my connection in between. So go to the particular question that we are looking at. We were here. So bacterial selection using antibiotic resistance gene. Yeah. So we are here bacterial selection using antibiotic resistance gene. So what we are talking about is how selection of specific bacterial colonies that have taken up your plasmid or are transformed with your plasmid are selected during experiments. What we do is that we have our plasmids carry a antibiotic resistance gene, just this, an ampicillin resistance gene. Now, if your bacteria has taken up your plasmid, then the bacteria will be ampicillin resistant. What we do is that we then go ahead and plate on a LB agar plate containing antibiotic ampicillin in it, the bacteria that you have transformed. So, if your bacteria is able to survive on the plate, then that means the selection has been done, and that is your bacteria contains your antibiotic resistant plasmid. So in this way you can shortlist or select bacteria which have taken up the particular plasmid that you require. So any other bacteria that have been contaminated in the plate cannot grow on the plate themselves. So this is a diagrammatic representation of what happens. Cells that do not take up the plasmid will not have antibiotic resistance, and when plated on a dish that contains an antibiotic, the cells will not survive. Whereas cells that have taken up the plasmid will have resistance to a specific antibiotic, and these cells will survive and proliferate on the cell, on the plate. So we will have colonies from the bacteria only that have the particular plasmid. Anyone else will not have any colonies on the plate. So from a given question, we have that the vector with that the gene of interest had a streptomycin resistant gene which was preserved after the cloning, but our vector's penicillin resistance gene was disrupted. So the transformed bacteria will not have a penicillin resistance because the gene was disrupted, but will have streptomycin resistance because the streptomycin resistance is present on the vector. So the correct option is the desired transformant will not have any penicillin resistance, that is, it will not survive in presence of penicillin, but it will survive in presence of streptomycin. So that was our entire discussion on the questions that were given throughout this the last year's course of week 2. So now we'll have a bit of discussion regarding uh, some other things that were discussed during the lectures. So the first thing that we want to discuss is blue-white screening. So what is blue-white screening? Blue-white screening is another selection method used in order to detect which particular plasmids have taken up your gene of interest and have been transformed into a bacteria. So say you have cloned your particular gene of interest into a plasmid vector and then transformed your bacteria with it. After transformation, if it so happens that the bacteria were only transformed with plasmids which did not have your gene of interest, so only the normal vector was transformed into the bacteria. So if you were screening using antibiotic selection, the plasmid has the antibiotic selection marker, and when you plate it, the bacteria will be well and good and happily survive on the plate. But this does not ascertain the fact that it will have your plasmid DNA inside it. So how will we detect the presence of a foreign DNA particle or your gene of interest in the vector backbone which is present in your transformed bacteria? So you want two things. First, that the plasmid is there, and two, that your gene of interest is present in the plasmid and present in the bacteria that is growing on your plate. So this is known as one of the techniques that are used for this is known as blue-white screening. So this is one step above antibiotic selection. So what you do is that you use a special type of E. coli strain, which is known as the competent E. coli strain. What happens here is the E. coli has the LAC-Z gene, but it which can convert beta galactosidase, like which breaks down beta galactosidase. So the LAC gene produces the enzyme beta galactosidase, which breaks down beta gal, and thus so this is the only function of the LAC-Z gene. But this has been used very cleverly in order to detect or screen bacteria, screen particular bacteria containing your plasma. So our competent E. coli has the LAC-Z, but a portion of it has been deleted. So this gene is known as the LAC-Z delta M13. What this means is that the beta galactosidase enzyme that will be produced by the competent E. coli will be inactive and will not cleave beta galactoside beta gal into its components. So that is there. But how does it help us? What happens is that your plasmid vector has its multiple cloning site present inside the other component of the LAC-Z known as LAC-Z alpha. So the deleted portion of the LAC-Z is present on the plasmid vector, and the multiple cloning site is present in middle of the LAC-Z. So what happens is that if your gene of interest is cloned in the vector, then the LAC-Z present in the plasmid vector will be disrupted. So if you have your gene of interest inside the plasmid vector, you will have your plasmid vector that does not contain a functional LAC-Z gene, but contains your gene of interest. In case there was no cloning done, then plasmid will have your functional LAC-Z but no insert, and that is this case. So now what will happen is that you want to go ahead and transform your this competent E. coli with the with the clone vector. So the clone vector will go inside the cell in all three cases. And now, if your LAC-Z present on the vector was not disrupted, that it does not have your required gene of interest, then the LAC-Z component from the plasmid vector will give, and the LAC-Z component from the E. coli cell individually will give rise to a functional beta galactosidase enzyme. So LAC-Z from the E. coli, LAC-Z from the vector combining together by a process known as alpha complementation will form a beta galactosidase functional enzyme. But in case your insert is present in the vector, then there is no question of a functional LAC-Z coming from the vector, and thus this colony which has your insert does not have a functional beta galactosidase enzyme. So in case we have our insert, no beta galactosidase. In case we do not have our insert, we have beta galactosidase. Now what you do is you take a, substrate known as X-gal, which upon action by beta galactosidase will produce color, which is produce a blue or indigo color, and a transducer or a inducer of beta galactosidase known as IPTG. And when you put both of them on the plate, what will happen is that functional beta galactosidase will go ahead and chop up the X-gal substrate, and after chopping the X-gal substrate up, it will produce blue colors, whereas your bacteria having your insert do not have any beta galactosidase, and they will not be able to cleave X-gal, and they will only have the white colonies in nature. And thus, via the entire process of screening, you will finally have a plate that looks something like this, with blue colonies and white colonies. And the white colonies will indicate the particular cells that have taken up your plasmid of interest, plasmid containing your gene of interest. So this is the process known as blue-white screening. And using this, you can just look at your plate and decide which cells which colonies are the positive clones, and you can go forward with those. So this was blue-white screening. If anyone has any question, they can post it. Okay, so we move forward, and the last discussion that we will have, it is on the lack of operon. So lack of operon is one of the most well studied operons present in bacteria, and they using this will mostly study how gene regulation works in bacteria. So the lac operon consists of a structural device such as this. We have a promoter site on which RNA polymerase binds. We have a cap site which is present upstream of the promoter site, which helps the binding of the RNA polymerase to the promoter. We have an operator site, and we have genes downstream of it. So the lac operon is inducible operon, which means in the normal condition it is in the off condition. So when it is what is the normal condition I'm talking about? That is when there is no lactose present in the medium. In that case, there is no requirement for the lac operon genes lac Z, lac Y, and lac A to be produced because there is no metabolism of lactose that is going on. So in that case, the lac operon is shut down, and the genes are not allowed to be transcribed or translated. So how is this regulation taking place? So there is a particular repressor that is present right here, which is expressed by the lac I gene. And this repressor, what it does is that it goes and binds to the operator region present between the promoter and the genes. So even if our RNA polymerase goes and binds to the promoter and wants to transcribe everything, the repressor acts as a physical barrier between the RNA polymerase and the genes, and the RNA polymerase cannot go ahead and bind to the and transcribe the genes, and thus the lac operon is suppressed or repressed, and thus we have no transcription or shut off of the lac operon. When lactose is absent, lac repressor binds tightly to the operator, and this prevents the RNA polymerase's way, preventing transcription. But in case of lactose, what happens is that after lactose metabolism, it forms something known as allolactose. Allolactose goes and binds to the repressor and does not allow the repressor to bind to the operator. And if there is no repressor binding to the operator, we have the RNA polymerase can happily go and bind to the promoter and then start transcribing the lac Z, lac Y, and lac A genes that are present in the lac operon, and thus we have switch on of the lac operon in presence of lactose. This is how the lac operon works. And there is also the question of the cap site. So we will discuss that also. So cap site binds to the cap protein. So Lacoperon has two stages of control. So first stage is what we can see right here in front of us. What the second stage occurs when there is no glucose present around the cell. When there is no glucose present around the cell, the cell by itself produces cyclic AMP, which is a metabolite. No glucose, we have cyclic AMP. Cyclic AMP goes and binds to the cap protein and allows the cap protein to tightly bind to the cap site present here. So we have a protein sitting on here. And this cap protein binding to the cap site is very helpful for the lab operon function. The cap protein, what it does is that it acts as an activator. What it does is that it helps RNA polymerase to bind to the promoter. And since RNA polymerase can actively bind to the promoter with the help of the cap protein, the rate of transcription is heavily increased, and this entire process increases the efficiency of the lac operon in the absence of glucose. If glucose is on the other hand present, but what happens is that there is no CAMP or cyclic AMP, no cap binding to the cap site, and RNA polymerase binding to the promoter is very poor in nature. and that is why we have low levels of operation of lac operon if there is glucose and lactose present. So even though lactose is present and the repressor is off, and the lac operon should be switched on, because of the presence of glucose, we have a secondary control over the lac operon and it does not act efficiently, and we have only lowered level of transcription from the lac operon. The major reason behind this is that lac operon, or the major reason behind this is that the preferred source of energy for E. coli or any other bacteria is glucose. So if there is glucose and lactose both present, it will prefer to have its glucose metabolism genes upregulated and lactose ones downregulated, so that it can preferably feed on glucose. And that is why in presence of glucose, lac operon is downregulated, or the genes being expressed in the lac operon are downregulated. So if we have no questions regarding this, I will bid you thank you for joining the session and watching the session throughout. In this particular session, we looked at genetic engineering, cloning, restriction enzyme digestion, PCR, and also talked about screening and selection of bacteria, transformation. So we were just wrapping up. In case you have any question, Pavitra, you can go ahead and unmute yourself and ask anything. So in this particular week, we talked about restriction digestion, cloning, transformation, PCR, and antibiotic selection and screening, as well as the lac operon. If you have any questions regarding this, you can go ahead and put it into the chat box or unmute yourself and ask anything. Okay, so maybe you can go ahead and watch the video on YouTube, and then you can get back to the to us with any question via the NPTEL questions portal that is there on the course site. So if we have nothing else to discuss today, we'll bid everyone thank you for joining the lesson, and see you all next week. Thank you.